Previously, we talked about taking this protein and unraveling it and beginning to look at the amino acid primary sequence of this protein. And in the last video, we talked about Edmonds reagent, and we said that Edmonds reagent will take this first amino acid out here in the beginning and it cleaves it off. And this lets us know what amino acid is in the front end of the protein. Well, if we've got a reagent that focuses on the N-terminus, then we also have a reagent that focuses on the C-terminus as well. And this reagent is called carboxypeptidase. So let me spell that. Carboxypeptidase. Carboxypeptidase. Now, out of the carboxypeptidases, there are a couple of different ones. We're going to be focused on carboxypeptidase A. Now, carboxypeptidase A cleaves off the C-terminus amino acid as long as it is not arginine or lysine. So that's the caveat here. So this will not work if it's arginine or if it's lysine. If I wanted the three-letter abbreviations for arginine, that is ARG, and R is the one letter. And then if I wanted the three-letter abbreviation for lysine, that is LYS, and that's a weird one, that's a K. Note that both of these go into the same family, which is why they're both listed. That would make sense. And these are your basic amino acids. So these are your amino acids that had that NH3 group or NH2 positive group that's on the R for the side chain. So very important, that's why they're both listed, but carboxypeptidase A will not work if there's arginine or lysine on the end. Now, we have a version of this carboxypeptidase. So carboxypeptidase B will only work and will only cleave if there's arginine or lysine at the end. Okay, so this kind of allows us to play both sides of the fence. If we take this protein and we add carboxypeptidase A to it, and we get an amino acid that gets shot off, so any of the other 18 amino acids will work, they all will get cleaved. That amino acid gets kicked off and we can analyze that using some kind of instrument or some kind of identification purpose, right? However, if we use carboxypeptidase A and we do not get an amino acid at all, that probably means that we have arginine or lysine at the end. And we are not seeing a cleavage that's happening at that end amino acid. So what we do then is that we throw in carboxypeptidase B. And then if it is arginine or lysine, we should see a cleavage point that begins to happen, and arginine or lysine will be kicked off, and the instrument will tell us which one it was, and then we can go on about the other protein sequencing after that point. So we do have two versions of carboxypeptidase. Now, there's actually other versions of carboxypeptidase that's out there, but A and B are the most common. So A cleaves it all off as long as it's not arginine or lysine, and B only cleaves if it's arginine or lysine. So very, very important. Now, there's no really structures here, so I'm going to put no structures that you got to know or memorize. And the reason is because these carboxypeptidases, they are enzymes. So they're actually proteins in a way themselves that do a specific job. So you don't have to memorize what these structures look like. You don't have to go through and figure out which one's which. But carboxypeptidase A and carboxypeptidase B, you have to know at least the differences between them. Why would I use one over the other? What is the difference when I add them to a solution of a protein? What do I get from one versus the other? So now there's three reagents that we need to look at when we look at protein sequencing for now. One is Edmonds that rips off the first one. One is carboxypeptidase that rips off the last one. And if carboxypeptidase A works, it will let you know this is the amino acid that I got. And then if it doesn't, we have to use carboxypeptidase B, and then we get arginine or lysine in the very end. Okay, with that said, we then can go through, once we identify the end and C terminus, and do something that we call hydrolysis. And hydrolysis is going to kind of do what we had mentioned before. It takes all of these amino acids and it frees them up from each other. We call this partial hydrolysis. And the reason that this is slightly different is because maybe we don't want to break up the entire protein. Maybe we only want some of the actual peptide broken up into the pieces. And this is what we call partial hydrolysis. So maybe this piece would have 3 amino acids in it. And maybe this piece would maybe have 5 amino acids in it. But it at least will take me a look at this protein, and it will let me break it down into bigger chunks. And these chunks I can actually go through and take a look and look at them a little bit closer, try to identify the chunks a little bit better, but that is what we call partial hydrolysis. How do we separate the fragments from each other? Well, we use something like electrophoresis or an amino acid analyzer. So we've talked about those before in the past. All of those are coming back. You can kind of see why those were important to mention very early on. So this is just a way that we can go through and take a look at the polypeptide and then hydrolyze it a little bit, not put it in such high acid content, not put it in such heat content, not keep it there for a long period of time, but at least put it in there, get it exposed, and allow it to break up a little bit better. So that is what we're after. That's what we want to do. And in the next video, we'll come back and we'll take a look at an example problem of how we can put all of this information now together into one kind of scenario that I will call mind games, because that's exactly what they are. They are mind games, little pieces and little puzzles that you're going to have to figure out how everything goes together accordingly. So in the next video, that's what we'll look at. So stay tuned. An example problem is coming. You might like it. You might actually have fun with it, or you might hate it, and you'll be like, there's no way I'm going to do these problems. So we'll see when you come back.